The one thing standing between us and curing genetic disease isn't science, it's size. In 2012, CRISPR-Cas9 detonated the world of biology. A molecular scalpel, guided by RNA, slicing into the genome with surgical precision. It wasn't just a discovery, it was a revolution, and it's already saving lives. In 2023, the FDA approved Casgevy, a CRISPR therapy for sickle cell disease and beta thalassemia. Patients' own cells, edited, reborn, and reinfused. But that victory came with an asterisk. These cells were edited outside the body. Safe, controlled, easy. However, the real war is being fought inside the body, in the liver, the eye, and the brain. And that's where CRISPR hits a wall. See, the Cas9 protein is massive. Almost 1,400 amino acids. A giant trying to squeeze through a door that isn't built for it. And in the world of therapy, that door is the AAV, the adeno-associated virus, biology's trusted delivery vehicle, already proven in landmark therapies like Luxturna and Zolgensma. But AAVs can carry only 4.7 kilobases. Therefore, the Cas9 protein alone is almost 4,500 bases, and therefore nearly all of that space is occupied by the Cas9 protein itself, which leaves almost nothing for the guide RNA it desperately needs to function. One way around this scientists use is to split it up. Two viruses racing to infect the same cell, hoping that both arrive in time. It's inefficient, it's fragile, and ultimately a gamble. So the hunts began for something smaller, something leaner, something that could fit. Enter Cas12f. Born not in bacteria but an ancient primitive archaea, a relic of the microbial world, barely 400 to 700 amino acids in length, a fraction of Cas9's size. But there's also a curse attached to this gift. Cas12f can fit, but it's much weaker than its Cas9 counterpart, until now. A new study published in Nature Structural Molecular Biology has fixed this problem. At just 500 to 600 amino acids, the entire editing machinery, including the enzyme, the guide RNA, every regulatory piece, finally fits inside a single AAV. One vector, one shot. And what's staggering is the efficiency. Over 80% editing efficiency in human cells, nearly 90% in some targets. A compact warrior, punching at heavyweight strength. This changes everything, because delivery, not editing power, has always been gene therapy's final boss. A single vector, compact editor could unlock tissues once thought untouchable, muscle, liver, even the retina, potentially even the central nervous system, guarded by the nearly impenetrable blood-brain barrier. This is no longer about proving CRISPR can work, it's about delivering it, into the body, into the cell, into the cure. We built the scalpel, now finally. If you like this kind of content, guys, please like and subscribe, and I'll see you in the next one.